Welcome to Bedtime Science. Did you know that the brains of men and women sometimes store the same moment in completely different ways, encoding the emotion in one mind and the detail in another? Tonight we look at the quiet, hidden science of how biological sex shapes the way memories form, linger, and fade inside the human brain. In the next moments, we will gently discuss how the hippocampus and amygdala encode memories differently in male and female brains, why emotional events tend to leave stronger traces in certain neural pathways depending on biological sex, how hormones like estrogen and testosterone quietly shape the way the brain holds onto the past, and what the hours you spend asleep do to help your brain sort, strengthen, and sometimes reshape the memories formed during the day. Before we begin, if you find comfort here tonight, consider subscribing to join our quiet community. And if you are still awake, you can leave a soft comment about what topic we should explore next. Now let's begin. Deep inside your temporal lobe, tucked beneath folds of cortex, there is a small curved structure shaped like a seahorse. It's called the hippocampus, and it is the place where new memories take their first breath. Men tend to have a larger hippocampus when you measure it in raw cubic millimeters. But here's what matters. That apparent advantage disappears entirely once you correct for total brain volume, because male brains are physically larger overall. After that correction, there's no statistically meaningful sex difference in relative hippocampal size. What does differ is the internal geography. Women tend to have a proportionally larger posterior hippocampus, the rear portion of the structure, a region strongly tied to spatial navigation and the mapping of places. So even when the overall volume looks equivalent, the internal architecture tells a quieter, more complicated story. Think of it as two houses with the same square footage but different floor plans. The rooms are arranged differently, and those differences in arrangement shape how each brain sorts and stores the world. The hippocampus doesn't work in isolation. It's wired into a sprawling web of connections that reach across both hemispheres. And those connections differ between men and women in ways that neuroscientists have been mapping for decades now. Women tend to show stronger connections between the left and right sides of the brain, a pattern scientists call interhemispheric connectivity. Men show stronger connections within a single hemisphere, particularly the left side. During spatial navigation tasks measured with functional brain imaging, women recruit more of the prefrontal cortex relative to the hippocampus. Men rely more heavily on the hippocampus itself. This pattern shows up not only in human brains, but also in rodent studies, where female rats display greater prefrontal involvement during maze tasks. These wiring differences don't make one brain superior. They suggest that male and female brains tend to favor different strategies to accomplish the same task, like two travelers reaching the same city by different highways. Now let's turn to a structure that sits just in front of the hippocampus. It's called the amygdala, a small, almond-shaped cluster of neurons that acts as the brain's emotional amplifier. When something happens that carries emotional weight, a startling noise, a painful loss, a moment of deep tenderness, the amygdala helps decide how deeply that experience will be inscribed into long-term memory. A landmark study from Canley, Desmond, Zhao, and Gabrieli, published in the Proceedings of the National Academy of Sciences in 2002, found something striking. When participants viewed emotionally intense images inside a brain scanner, The women who remembered those images most accurately showed the strongest activation in their left amygdala. The men who remembered best showed the strongest activation in their right amygdala. 72% of women showed this left-sided pattern. 50% of men showed the right-sided pattern. This isn't a minor anatomical footnote. It points to a fundamentally different neural organization governing how emotional salience becomes a durable memory in each sex. That same 2002 study revealed something even more subtle about how each brain links feeling to remembering. In women, the same amygdala, the left one, participated in both evaluating how emotional an experience felt and encoding that experience into memory. The researchers called this a concordant pattern, evaluation and storage happening under the same roof. In men, the process split apart. One amygdala handled the emotional evaluation, the other handled the encoding, a discordant pattern, two separate systems running two separate jobs. When the researchers searched for brain regions involved in both processes at once, they found 9 active regions in women, including the left amygdala, versus only 2 frontal regions in men, with no medial temporal involvement at all. This tighter coupling in women between how emotional something feels and how deeply it gets filed away may be one reason why emotional memories tend to leave such vivid, lasting traces in the female brain. Larry Cahill and colleagues confirmed these findings independently in a 2004 brain imaging study. Using a different set of emotional images and a different group of participants, they found the same hemispheric split, a stronger right amygdala to memory relationship in men, a stronger left amygdala to memory relationship in women. 2 laboratories, 2 different experimental designs, the same quiet asymmetry showing up in the data. That kind of replication, across independent studies with different methods, is what makes a finding robust in neuroscience. The behavioral side of this story is just as revealing. In the original 2002 study, women rated more pictures as highly emotionally arousing compared to men. And for those high-intensity images specifically, women showed better recognition memory, both in terms of feeling familiar with the images and in terms of genuinely remembering them. But here's the key nuance. For images rated as less emotionally intense, no sex difference in memory appeared at all. None. And the rate of false memories, incorrectly believing you'd seen something you hadn't, was nearly identical between the groups. About 12% for women, about 10% for men. So the difference isn't that women have better memory across the board. It's that for the most emotionally charged moments, the female brain encodes more deeply. A targeted amplification effect, not a blanket advantage. Men and women also differ in how they organize their personal histories. A 2016 study in Frontiers in Human Neuroscience asked participants to recall autobiographical memories, real events from their own lives, while lying inside a brain scanner. The researchers sorted these memories by personal importance, emotional tone, and how long ago they had happened. Men showed greater activation in the left parahippocampal region, a structure specialized for spatial context and scene construction. Women showed greater activation in the right dorsolateral prefrontal cortex, a region associated with temporal ordering, cognitive control, and verbal elaboration. In plain terms, men tended to organize their memories around where things happened. Women tended to organize them around when things happened, and what those moments felt like. Two different filing systems for the same life. That same study uncovered another layer. Women showed stronger activation in the right insular cortex, specifically when retrieving older memories and negative memories. The insula is a deeply folded region of cortex involved in interoception, the brain's ability to sense the internal state of the body. It helps generate the felt quality of experience, the heaviness in your chest during grief, the warmth of a happy recollection, the tightness that comes with worry. Women's stronger engagement of this region during autobiographical recall may explain why female memories often feel more bodily, more emotionally textured, more present in the senses, even when the objective accuracy of the memory is no different from a man's. It's not that women remember more facts, it's that they refeel more intensely while remembering. A meta-analysis led by Spetz and Slotnick reviewed 8 brain imaging studies of long-term memory retrieval and identified regions where men showed greater activation than women during recall. The lateral prefrontal cortex, visual processing regions, the parahippocampal cortex, the cerebellum. The researchers explicitly criticized the common practice of combining male and female data together in cognitive neuroscience studies, arguing that doing so hides real differences in how the two brains operate. The regions where men showed greater activation map onto visuospatial processing and scene-specific context retrieval. They fit neatly with the idea that men tend to use more visually based memory strategies, relying on spatial landmarks and environmental layout rather than emotional or verbal tags to anchor a memory. A 2024 study using functional near-infrared spectroscopy, a technology that measures blood oxygenation in the brain through light sensors placed on the scalp, examined working memory performance in men and women during a task called the NBAC. Women outperformed men on accuracy across all difficulty levels. The statistical effect was large. But here's what made the finding unusual. Women achieved this higher performance with less brain activation. They needed less cerebral blood oxygenation in the prefrontal cortex to get better results. Think of it as fuel efficiency. The female brain appeared to recruit working memory circuits more selectively, using fewer resources for equal or better outcomes. The male brain activated more broadly, with greater intensity for the same task. More activation doesn't always mean better performance. Sometimes it means the brain is working harder to reach the same destination. That same 2024 study uncovered a striking pattern in how the two hemispheres cooperated during working memory. In the left hemisphere, men showed stronger functional connectivity between prefrontal regions. In the right hemisphere, the pattern reversed completely. Women showed roughly 4 times greater connectivity than men, and that connectivity climbed as the memory task became harder. Left-dominant males, right-dominant females. Not just a difference in degree. A qualitative reversal in how the brain's two halves coordinate during the same cognitive operation. At the whole brain level, women have approximately 14% more local functional connectivity density and about 5% greater gray matter density than men, according to a large-scale neuroimaging analysis by Tomasi and Volkow. A denser, more locally interconnected cortex may be one reason women can achieve equivalent or better cognitive outcomes with less total activation. The architecture itself is more compact, more tightly woven, requiring fewer long-range signals to assemble a working network. It's like the difference between a compact neighborhood where everything you need is within walking distance and a sprawling suburb where you have to drive across town. Both arrangements get the job done. One just requires less travel. A meta-analysis drawing on the BrainMap database, one of the largest repositories of brain imaging data in the world, examined which neural networks men and women activate during working memory tasks. Women consistently activated more limbic structures, the amygdala on both sides, the right hippocampus, the anterior cingulate cortex. These are regions tied to emotional processing and contextual memory. Men activated a different collection of regions: the cerebellum, the superior parietal lobe, the left insula, the thalamus on both sides, regions linked to spatial attention and sensory motor integration. The implication is subtle but worth sitting with. Women appear to weave in emotional context even during tasks that have no emotional content at all. Men deploy a more purely spatial attentional backend, two different engines running under the hood during the same mental operation. In 2024, a team of researchers trained a deep neural network to tell male and female brains apart using resting state brain scans. Not scans taken during a task, scans taken while people simply lay still, doing nothing at all. The network achieved classification accuracy above 90%, and it worked across 3 independent datasets involving more than 1,000 participants. When the researchers used explainable artificial intelligence to trace what the network was picking up on, the strongest signals came from the default mode network, the striatum, and the limbic network. The default mode network, which is active during mind-wandering, self-reflection, and memory retrieval, showed the largest effect sizes across all datasets. Sex differences in memory-relevant brain networks are present even at rest, even when the brain is doing nothing in particular. The same deep learning study went one step further. It asked whether the brain features that distinguished male from female brains could also predict cognitive abilities. In women, the most distinctive brain features, located in the ventromedial prefrontal cortex, the middle temporal gyrus, the posterior cingulate, and the precuneus, correlated with general intelligence. In men, the distinctive features in the dorsolateral prefrontal cortex, the posterior cingulate, and the postcentral gyrus correlated with reward sensitivity and delay discounting, the tendency to prefer immediate rewards over delayed ones. And when the researchers tried applying one sex's brain model to the other, prediction dropped to chance. The female model could not forecast male cognition. The male model could not forecast female cognition. Two brains, two cognitive signatures, each one legible only in its own biological language. Resting state connectivity between the left and right hippocampus, measured within the default mode network, is markedly greater in women than in men, according to a 2024 study in cerebral cortex. This enhanced bilateral synchrony at rest may help women integrate contextual information more rapidly during memory encoding, and it may support richer cross-hemispheric consolidation during sleep. But after menopause, when estrogen levels fall, this connectivity pattern changes. Postmenopausal women lose the normal ability to suppress this resting connectivity during active memory encoding. The brain fails to shift gears, and memory performance drops. Estrogen, it turns out, doesn't just build synapses, it maintains the flexible regulation of entire networks. The development of these memory structures is neither steady nor symmetrical between boys and girls. The hippocampus and amygdala tend to increase in volume during childhood and early adolescence, then begin to decline in late adolescence. But the timing of this arc differs between the sexes, shaped by the surge of gonadal hormones that arrives with puberty. There's also a right hippocampal asymmetry in childhood. The right hippocampus is typically larger than the left, and this interacts with sex differences in the pace of structural maturation. Because hippocampal development responds directly to both estrogen and testosterone, the divergence between male and female memory profiles that emerges through the teenage years reflects, in part, the physical remodeling of these circuits under hormonal influence. Your brain at 15 is being actively reshaped by chemistry. Even in the pathology of Alzheimer's disease, sex differences in hippocampal architecture shape how neurodegeneration unfolds. In Alzheimer's patients, elevated levels of neurofibrillary tangles are associated with reduced neuronal density in different hippocampal subregions depending on sex. In women, the CA1 region takes the hardest hit. In men, the CA3 region bears the brunt. Total tau and amyloid beta levels in women are associated with steeper cognitive decline and faster hippocampal volume loss compared to men, effects that become especially pronounced in women carrying the APOE4 risk allele. The female and male hippocampus are not simply scaled versions of each other. They have different cellular compositions, and those compositions create different weak points when disease arrives. Large-scale lifespan brain imaging studies have found that men show greater age-related cortical thinning than women in several regions directly relevant to memory. The cuneus, the lingual gyrus, the parahippocampal cortex, the paracalcarine cortex. These regions support visual scene processing, spatial context, and the bridge between seeing and remembering. So while women carry the greater burden of Alzheimer's at a population level, men appear more vulnerable to thinning in the circuits that support visuospatial memory specifically. Healthy aging affects the two sexes through anatomically distinct paths, different regions, different timelines, different consequences for how your world gets stored. The hemispheric asymmetry in working memory deserves one more look. During baseline functional connectivity measurements, men exhibit stronger left hemisphere connectivity, while women exhibit stronger right hemisphere connectivity. And the interaction between sex and task load was statistically measurable in at least 9 separate channel pairs. As working memory load increased, the pattern of connectivity change moved in opposite directions for the two sexes, left versus right, up versus down, not merely a quantitative difference but a qualitative reversal. This represents one of the clearest demonstrations in neuroimaging that female and male working memory circuits are mirror opposite in their hemispheric organization. Now let's move beneath the architecture and into the chemistry. Estrogen is one of the most powerful natural modulators of hippocampal structure in the female brain. It doesn't merely support memory. It physically reshapes the circuits where memories form. In the hippocampus, estrogen triggers spinogenesis, the growth of new dendritic spines, those tiny protrusions on neurons where synaptic connections are made. It also promotes synaptogenesis, the formation of entirely new synaptic contacts. This effect is not slow. not gradual. Estrogen can reshape the physical architecture of memory circuits within hours of exposure. Female rats at peak estrogen, during the prostrus phase of their reproductive cycle, show greater dendritic spine density in the CA1 region of the hippocampus compared to both males and low estrogen females. More spines, more contact sites, more opportunities for incoming information to be captured and held. The molecular pathway behind this is specific enough to trace step by step. Estrogen stimulates the formation of filopodia, thin, finger-like protrusions that are the precursors to mature dendritic spines. It does this through a cascade involving a protein called LIMK1, which phosphorylates and inactivates another protein called cofulin. Cofulin normally severs actin filaments, the structural scaffolding inside neurons. By switching off cofulin, estrogen frees actin monomers to polymerize into stable, spine-forming structures. A direct molecular link, from a circulating hormone to a physical expansion of the synapse. This pathway carries clinical weight, too, because disruption of actin dynamics is increasingly recognized as a feature of both normal aging and Alzheimer's pathology. When this scaffolding system fails, the architecture of memory quietly crumbles. Estrogen also boosts the expression and binding activity of NMDA receptors in the hippocampus. NMDA receptors are the principal molecular switches for synaptic plasticity, the process by which connections between neurons grow stronger with use. When researchers pharmacologically blocked NMDA receptors, estrogen could no longer trigger dendritic spine formation in the CA1 region. The structural response needed the receptor to be active downstream of the hormone signal. This connects the hormonal fluctuations of the menstrual cycle directly to the molecular machinery of learning. Every month, as estrogen rises and falls, the ability of hippocampal synapses to strengthen and grow shifts with it. The interplay between estrogen and progesterone at the synapse is more complex than you might guess. After estrogen has elevated dendritic spine density in the CA1 region, a short exposure to progesterone produces a brief additional boost, lasting roughly 6 hours. A small enhancement window opens. And then something counterintuitive happens. Progesterone triggers a rapid and substantial reduction in spine density, dropping it back toward or even below baseline. This rebound pruning may serve a homeostatic purpose, preventing runaway synaptic expansion. But it also means that the female brain's synaptic architecture undergoes a cyclical expansion and contraction pattern across the menstrual cycle. The architecture is not fixed. It breathes. It pulses with hormonal rhythm. Estrogen acts through at least 2 distinct receptor subtypes in the brain, Rα and Rβ, and they do different things in different places. Rβ is the most common estrogen receptor in the hippocampus and is specifically linked to learning, memory, and spatial navigation. Rα is more prominent in hypothalamic and limbic circuits. Here's the thing. A 2024 study found that Rα interacts directly with tau protein, the pathological aggregate found in Alzheimer's disease. Tau can sequester Rα, pulling it away from the cell nucleus and stripping the neuron of its estrogen-responsive neuroprotective signaling. The very disease that threatens memory also dismantles the system designed to defend it. Testosterone shapes memory too, though through a different set of circuits. Studies in male rodents have shown that removing the primary source of testosterone impairs spatial learning and memory in the Morris water maze. Testosterone replacement restores performance, but the effect isn't instant. It requires at least 7 days to take hold, suggesting a slower structural reorganization rather than a quick pharmacological kick. The effect follows a dose-response curve. More testosterone, better spatial memory, up to a point. This pattern is consistent with the gradual decline in spatial cognition that men experience as endogenous testosterone falls with aging. In humans, a randomized, double-blind, placebo-controlled trial published in Neurology in 2001 by Janowski and colleagues gave 25 healthy men between the ages of 50 and 80 either weekly testosterone injections or a saline placebo for 6 weeks. Testosterone levels rose by 130% in the treatment group. Improvements appeared in spatial memory, recalling a walking route, as well as spatial ability and verbal memory. But there was a wrinkle. Because testosterone is partly converted to estrogen through a process called aromatization, estradiol levels also climbed by about 77%. So the cognitive benefits may have come from testosterone directly, from its conversion to estrogen, or from both working together. The two hormone systems are deeply intertwined. Even in the male brain, estrogen plays a quiet supporting role. Progesterone's influence extends beyond the synapse into the stress hormone system. A study of 56 women compared those in the hyprogesterone mid-luteal phase of their cycle with those in a low progesterone phase. Both groups viewed neutral and threatening images, and then either received a stress challenge or a control condition. Women with high progesterone showed higher baseline cortisol and greater stress-evoked cortisol. And critically, they showed enhanced memory for negative images when stress was present. A positive correlation emerged between stress-induced cortisol and recall of threatening images. Your hormonal phase can shape which memories stick most tenaciously after a stressful event. Cortisol itself, the primary stress hormone, affects memory through mechanisms that are fundamentally different in each sex. In young men, a cortisol increase during memory tasks showed a strong negative correlation with memory performance. The correlation coefficient was -0.82. That's a tight, almost linear relationship. High cortisol, poor memory. In young women, no such association appeared. In middle-aged populations, the pattern shifted. Only women showed an acute stress impact on memory, and in postmenopausal women specifically, higher cortisol was associated with poorer recall. The male hippocampus appears more acutely vulnerable to cortisol elevation. The female hippocampus, especially in reproductive age women with intact estrogen, shows a kind of resilience to acute cortisol interference. But menopause strips that buffer away. At the level of individual synapses, the molecular machinery of learning itself works differently in the two sexes. In a circuit connecting the hippocampus to the nucleus accumbens, a pathway linking memory to reward-motivated behavior, both sexes achieve equivalent long-term potentiation. The outcome is the same, but the mechanisms underneath, completely different. In females, long-term potentiation in this pathway requires L-type voltage-gated calcium channels and the activity of estrogen receptor alpha. In males, it requires NMDA receptors and does not depend on estrogen receptor alpha at all. Both sexes converge on the same downstream molecular step, a protein called CAMKII, but they arrive there by entirely different routes. Consider what this means for medicine. A drug targeting NMDA receptors to rescue synaptic plasticity might work in males but fail in females. And a drug targeting L-type calcium channels might work in females but fail in males. Two brains, two lock-and-key systems for the same cognitive function. At the CA1 synapse specifically, female rats show larger baseline excitatory responses mediated by AMPA receptors compared to males. Their synapses are already running closer to full strength at rest. That sounds like an advantage, but it creates a paradox. Because the synapse is already near its ceiling, there's less room for further strengthening during learning. Males, starting from a lower baseline, show greater long-term potentiation magnitude, a wider dynamic range for change. The mechanism involves a signaling pathway using cyclic GMP and protein kinase G, which drives the insertion of additional AMPA receptors into the postsynaptic membrane during potentiation. Males show enhanced activity in this pathway. So the two sexes operate their hippocampal synapses at different set points along the plasticity spectrum. Females with higher resting strength, males with greater room to grow. Pharmacological experiments have dissected this further with clean, reciprocal results. Blocking a specific subtype of NMDA receptor, called GLU-NTB, eliminates long-term potentiation consolidation in males only. Blocking estrogen receptor alpha eliminates it in females only. Each intervention knocking out plasticity in exactly one sex. These results carry heavy implications for how we interpret decades of preclinical research on memory and learning, most of which was conducted using only male rodents. Brain-derived neurotrophic factor, or BDNF, is the most important growth factor for synaptic plasticity and neuronal survival in the adult brain, and it shows consistent sex differences in expression. Adult female rats have higher BDNF content than males in the hippocampus, the hypothalamus, the cortex, and the amygdala. Estrogen positively regulates BDNF gene expression, providing a direct hormonal mechanism for this pattern. Enriched environments, which are known to boost BDNF, produce even greater increases in females than in males, particularly in the prefrontal cortex and hippocampus. Female brains may be more responsive to environmental enrichment as a memory-boosting intervention. But here's an interesting developmental twist. In early life, the pattern is reversed. Neonatal male rodents have roughly 50% more BDNF transcripts in the dentate gyrus and CA1 compared to females. Males start ahead. By adulthood, females pull ahead. A crossover that parallels the developmental timeline of many behavioral sex differences in memory. Oxytocin, sometimes called the bonding hormone, also plays a role in memory, and it plays that role differently in each sex. Oxytocin improves recall of emotional faces in women, but in male macaques, it enhances working memory specifically. The same molecule entering different neural circuits, producing different cognitive effects depending on the sex of the brain receiving it. A 2016 study published in PNAS found that oxytocin administration produces opposite amygdala responses when men and women receive social feedback. In women, oxytocin increased amygdala activation while hearing praise. In men, It increased amygdala activation while hearing criticism. Oxytocin amplifies what's motivationally relevant for each sex. Bonding-related signals for women, status-related challenge for men, and whatever gets amplified in the amygdala gets encoded more deeply into memory. Allopregnanolone, a neuroactive steroid derived from progesterone, enhances a type of brain inhibition mediated by GABA receptors, and it has neuroprotective properties that differ by sex. Female brains show greater sensitivity to allopregnanolone-mediated GABA potentiation, and allopregnanolone provides greater protection against excitotoxic and ischemic insults, the kind of damage that occurs during strokes in females than in males. This sex-specific neuroprotective advantage may partly explain why women experience lower rates of stroke-induced memory loss compared to men, even accounting for differences in body size and head trauma patterns. But because allopregnanolone levels fluctuate with the menstrual cycle, peaking in the luteal phase when progesterone is high, this protection waxes and wanes with hormonal rhythm. It's not a constant shield, it's a cyclical one. One more detail about calcium and synaptic learning before we leave the chemistry behind. Females rely primarily on L-type voltage-gated calcium channels as the trigger for long-term potentiation in certain hippocampal pathways. Males rely on NMDA, receptor-mediated calcium influx. L-type channels open when the cell membrane depolarizes. They have a lower threshold. NMDA receptors demand both ligand binding and depolarization at the same time, a stricter coincidence detection system. The female reliance on L-type channels may make female synapses more responsive to strong but temporally imprecise inputs. The male NMDA dependence implements a tighter Hebbian learning rule, where timing matters more. These different calcium sources also respond differently to aging and disease, with consequences for sex-specific vulnerability to synaptic failure. Glucocorticoid receptor expression in the hippocampus, the very receptors through which cortisol acts on memory, declines with age. But the decline isn't equal. Post-mortem analysis of hippocampal tissue found that women show a measurable reduction in glucocorticoid receptor expression with aging. Men show only a weak, non-significant trend. As these receptors disappear from the female hippocampus, the protective buffering that younger women enjoy gradually erodes. Declining estrogen, declining receptor expression, increasing cortisol sensitivity. These changes compound across the decades of postmenopausal life, each one making the next one worse. And at the receptor level, the ratio of AMPA to NMDA receptors at the CA1 synapse is higher in females than males. A higher ratio generally reflects a more mature, already potentiated synapse. But even the specific estrogen receptor that mediates synaptic enhancement differs by sex. In males, beta handles the job. In females, a different receptor called GPER, the G-protein-coupled estrogen receptor, takes the lead. Both converge on the same strengthening of the synapse, but through different molecular doors. Let's shift now from molecules to memories, the kind you carry with you, the kind that make up the story of your life. Behavioral studies of autobiographical memory consistently find that women report more vivid personal memories, more details about what emotions they experienced during the event, more awareness of what other people present were feeling, more sensory qualities, sounds, textures, temperatures, and a stronger sense of personal meaningfulness. Men tend to report memories that are more factual, more spatially oriented, more focused on what happened and where, rather than what it felt like. These differences aren't merely reporting biases. They track the neural differences in how the insular cortex, the dorsolateral prefrontal cortex, and the amygdala are recruited during recall. One theoretical account of this is the affect intensity hypothesis. The idea is straightforward. Women experience events with greater emotional intensity at the moment they happen, and that greater intensity drives deeper processing and richer encoding into long-term memory. Brain imaging data supports this. Women show greater bilateral anterior temporal activation and right limbic activation during autobiographical recall, consistent with stronger emotional re-experiencing. If the emotion is felt more deeply at the time of encoding, the memory trace it leaves will naturally carry more detail. Not because the filing system works differently in some abstract way, but because it was given richer raw material to work with. A complementary account is the cognitive style hypothesis. This one proposes that men preferentially organize memories using spatial context, recruiting the parahippocampal cortex for scene-based and landmark-referenced recall. Women use temporal context as their organizing principle, sequencing memories along a timeline, tagging them with emotional markers and social landmarks. The neural evidence lines up: parahippocampal activation in men, dorsolateral prefrontal activation in women. These cognitive style differences could emerge from hormonal organization during development, from culturally reinforced habits, or from some combination of both that's difficult to untangle. And here's something worth sitting with for a moment. Despite all these neural differences in encoding and retrieval strategies, direct comparisons of behavioral memory performance between men and women in autobiographical memory studies typically find no measurable group differences. Objective accuracy is comparable. Emotional intensity ratings are comparable, even though the brain activation patterns underneath are substantially different. Different brains, same outcome. Neuroscientists call this equifinality. Multiple routes leading to the same destination, with different strengths and weaknesses that only reveal themselves under unusual conditions. Women are faster and more accurate at recognizing all types of emotional facial expressions, including happiness, surprise, fear, anger, disgust, and sadness, as well as neutral faces. A 2018 study confirmed this advantage across every category tested. The reaction time differences were robust. They replicated across multiple stimulus sets. Faster emotional appraisal means stronger amygdala activation, and stronger amygdala activation drives deeper encoding. The speed of perception feeds directly into the depth of memory. Even more notable, this advantage persists when conscious visual processing is disrupted. Using a technique called continuous flash suppression, which prevents participants from consciously seeing the images, women were still faster to detect emotional expressions breaking through the suppression. The female advantage in emotional face recognition isn't solely a conscious strategy. It reflects a more automatic, preconscious sensitivity in the emotion perception system. The brain is tagging emotional stimuli as important before the person even realizes they've seen them. And if your brain tags something as significant before you're even aware of it, The memory trace starts forming at a level below conscious control. Women also show a broader advantage in face recognition memory, linked in part to verbal encoding strategies. Women are more likely to spontaneously generate verbal labels and social categorizations for faces: a name, a personality impression, a social category. This strengthens the memory trace through multimodal encoding. Both men and women show better recognition of same-sex faces, but this own-sex bias is larger and more consistent in women, suggesting greater investment of attentional resources in encoding female faces, possibly reflecting stronger same-sex social monitoring. Across multiple studies, women spontaneously generate and recall more negative personal memories in free recall tasks. This pattern shows up in both young and older adults. The tendency to recall more negative memories is likely driven by the same left amygdala circuit that amplifies emotional encoding, combined with more frequent spontaneous rumination about difficult experiences. This isn't a flaw. It's an optimization for emotional learning that carries costs in mood regulation. The same system that helps you learn deeply from painful experiences can keep replaying them long after they're useful. Women show greater dorsolateral prefrontal cortex activation, specifically for positive memories. In men, this activation isn't modulated by emotional valence. It's applied uniformly regardless of whether the memory is happy or sad. The dorsolateral prefrontal cortex is a key region for cognitive control. Women's greater engagement of this region for positive memories may reflect an active process of holding on to and elaborating pleasant experiences through prefrontal control, not passive replay, active savoring. These neural differences in autobiographical recall aren't limited to recent events. They persist across the full time span of personal history. The female insular activation pattern shows up for old memories and new ones alike. The male parahippocampal pattern is equally consistent across memory age. Whatever strategies each brain uses to organize personal history, those strategies endure over decades. Whether you're remembering something from last Tuesday or from childhood, the same neural architecture shapes the retrieval. Women's autobiographical memories tend to be what researchers call episodically thick, richer in information about who was present, what was said, what emotional states circulated in the room, what the social relationships involved implied. Men's memories tend to center more on what they personally did, their own actions, their own decisions, their own outcomes. This distinction has practical consequences in areas as varied as eyewitness testimony, couples communication, and therapeutic recall during psychotherapy. Stress interacts with emotional memory differently in each sex. Under stress conditions, women show enhanced recall of negative images. Men show enhanced recall of neutral images. Stress-induced cortisol predicted negative image recall in women and neutral image recall in men. This crossover may partly explain the higher rates of post-traumatic stress disorder in women. Stress hormones selectively amplify the encoding of threat-relevant material specifically in the female brain, while the male brain under stress seems to sharpen its focus on baseline, non-emotional information. Women also recall the physical appearance of people they've interacted with more accurately than men, under both directed and incidental learning conditions. Not just faces, but the whole physical presentation. This reflects a broader female orientation toward tracking social information in the environment, monitoring the signals of others' states, status, intentions. That monitoring translates into richer encoding of person-specific details that men tend to overlook or discard. While women excel at explicit recognition of emotional faces and social memory, men show a different pattern with emotional material. When men do engage with emotional content, the contrast between emotional and neutral material is particularly strong in their memory. The signal-to-noise ratio of emotion versus neutrality is higher. Men may process a narrower but more distinctly filed emotional category. Women integrate emotional context more uniformly across all material. Two different relationships between feeling and remembering. From an evolutionary perspective, the pattern makes a kind of quiet sense. Enhanced female memory for social signals, kinship relationships, emotional states, and interpersonal histories would have supported fitness in cooperative social environments where managing complex relationships was essential. Enhanced male spatial and route memory would have supported navigation, territorial awareness, tracking prey across unfamiliar ground. How much of this is biologically fixed versus culturally amplified remains an open question. But the neural architecture underlying it is demonstrably present in the adult brain. Now let's look at the large-scale behavioral evidence. A 2019 meta-analysis by Asperholm and colleagues, synthesizing 617 studies, confirmed that women outperform men on episodic memory tasks. But the magnitude of the effect depends critically on what type of material was tested. The female advantage is largest for verbal material, word lists, paired associates, stories. It extends to nameable images and object locations. The male advantage appears for abstract, non-nameable images and spatial routes. This material type dependency isn't a statistical confound. It's a mechanistic fingerprint. The female verbal emotional encoding system excels with material that can be labeled with language. The male visuospatial system handles abstract patterns and route-based navigation more efficiently. A clean way to think about it. Women's episodic memory advantage extends to verbal tasks, pictures with nameable content where verbal labeling is possible, and object location memory. Men's advantage covers abstract images where verbal encoding doesn't help, and spatial route memory, where landmark-free navigation strategies work best. This double dissociation maps onto the hypothesized left hemisphere verbal labeling strategy for women and right hemisphere visuospatial strategy for men. It predicts that any manipulation preventing verbal encoding, like articulatory suppression, should narrow or eliminate the female advantage while leaving the male advantage intact. A 2022 meta-analysis in Perspectives on Psychological Science examined 496 effect sizes across 50 years of data and found that women consistently outperform men in phonemic verbal fluency, generating words beginning with a given letter, with an effect size of about 0.12 to 0.13. In episodic recall tasks, the female advantage was about 0.28. The stability of these numbers across 5 decades, through changing educational systems, shifting gender roles, evolving testing environments, is worth noting. The female verbal memory advantage is not an artifact of cultural factors that have since equalized. On the other side, the male advantage in three-dimensional mental rotation is one of the largest cognitive sex differences ever documented. The effect size runs from about 0.8 to 1 on the Cohen's d scale, comparable in magnitude to the sex difference in physical height. This advantage has been demonstrated in children, adolescents, adults, the elderly, in paper and pencil tests and computer-based tasks, across dozens of cultural contexts. How universal is it? Silverman and colleagues tested 244,893 participants across 40 nations and found a meaningful male advantage in mental rotation in every single one. Every nation tested, regardless of language, educational system, or gender equality index. The cross-national universality suggests a biological substrate that can't be fully explained by socialization or cultural exposure. But the size of the advantage varies across cultures, meaning cultural factors modulate the difference without creating it from scratch. The developmental timing of these differences offers another clue. Verbal fluency sex differences emerge in childhood, with girls outperforming boys even in early schooling. Spatial ability differences are typically small or absent in young children and magnify substantially during and after puberty. This pattern directly points to gonadal hormones, rising testosterone in males, rising estrogen in females, as triggers for the spatial divergence. Studies have confirmed this by showing that testosterone supplementation improves spatial performance in hypogonadal adolescents. Fine-grained developmental data shows girls score higher than boys on verbal comprehension at ages 2 through 4. This aligns with girls' earlier language onset, larger early vocabulary, faster sentence development. But by ages 10 to 11, boys catch up and overtake girls on the same measures. The crossover corresponds approximately to the onset of male puberty and the increasing contribution of visuospatial organization to complex verbal reasoning. The story of women being better at verbal memory is most accurate in childhood and adulthood. Around puberty, the picture briefly inverts. An important mechanistic finding: after statistically controlling for mental rotation test scores, sex differences in spatial working memory tasks largely disappear. This suggests the male advantage in spatial working memory isn't an independent, primary sex difference. It's a downstream consequence of mental rotation ability. The same neural hardware that supports three-dimensional object manipulation also supports the maintenance of spatial information in working memory. Training mental rotation might selectively reduce the spatial working memory gap between the sexes. Not all spatial abilities show the same degree of sex difference. Large-scale spatial ability, navigation in real environments, map reading, geographic orientation, shows an effect size of about 1.34. Small-scale spatial ability, tabletop mental rotation, spatial visualization on paper, shows 0.62, more than double the difference for real-world navigation. Real-world navigation engages the hippocampus and entorhinal cortex in ways that tabletop tasks can't fully replicate. And it's precisely in this domain, large-scale spatial memory, where GPS technology has most dramatically substituted for natural ability. In real-world exploration tasks, males exhibit more efficient navigation patterns and better incidental memory for the locations of objects encountered during exploration. Females explore more systematically relative to landmarks and show stronger memory for object identity and object-landmark associations. Two styles. Males extract spatial layout with fewer traversals, using an allocentric, map-based reference frame. Females build richer object location databases through more repeated, landmark-anchored exploration. Each approach has trade-offs that only become visible under specific testing conditions. The female advantage in object location memory isn't present in young children. Studies show it's not statistically measurable in children aged 4 through 10. The advantage develops gradually through adolescence and reaches its characteristic adult magnitude only after puberty. Again, this timeline points to estrogen-driven changes in hippocampal circuit organization, the spinogenesis and connectivity remodeling discussed earlier, as the biological substrate for this emerging ability. A 2026 study tracked spatial and texture imagery abilities across age groups and found that the sex difference in spatial imagery favoring males emerges specifically around ages 13 to 14, coinciding with the height of male puberty. Before that age, the difference is small or absent. Afterward, it persists robustly through adulthood. But the same study found that females outperform males consistently in texture imagery and shape imagery, tasks requiring fine-grained perceptual memory of surface properties and object forms, across all age groups tested, including children. This female advantage in object property memory is present before puberty and doesn't change at the pubertal transition. It seems to reflect an earlier set neural organization, perhaps influenced by prenatal hormone exposure, rather than a puberty-triggered shift. An often overlooked dimension of sex differences in memory is variance. Men show greater variance than women in verbal episodic memory and spatial location tasks. The male distribution is wider, with more individuals at both the very top and very bottom of the performance range. Women show greater variance for root memory. Greater male variance means that while the female mean is higher for verbal episodic memory, the single highest performing individuals are more likely to be male. A commonly misunderstood aspect of group differences in cognition. The relationship between spatial working memory, mental rotation, and real-world navigation forms a hierarchy. Superior male navigation is substantially explained by superior spatial working memory, which is in turn substantially explained by superior mental rotation. When you statistically control for these foundational abilities, the sex differences in higher-order navigation tasks shrink considerably. The male navigational advantage isn't an independent gift for geography. It cascades upward from a difference in how three-dimensional spatial information is processed and held in mind. Let's allow the topic to drift now, gently, toward what happens when you close your eyes at night. One of the most consistent sex differences in your nightly brain activity involves the distribution of time across different stages. Women consistently spend more time in slow-wave sleep, stage N3, the deepest, most restorative phase. Men spend proportionally more time in stages N1 and N2, the lighter phases. Women spend approximately 8 extra minutes in non-REM sleep overall. That may not sound like much on any given night, but slow-wave sleep is the stage during which declarative memories are preferentially consolidated through a dialogue between the hippocampus and the neocortex, mediated by sharp wave ripples and slow oscillations. 8 extra minutes in the phase that matters most for memory consolidation, repeated every single night across a lifetime. It accumulates. While women dominate in slow-wave sleep, the picture changes during REM. REM duration tends to be longer in males across most of the adult lifespan. Older postmenopausal women may eventually exceed their age-matched male counterparts in REM duration, but for most of adulthood, men get more REM. REM sleep is linked to procedural memory consolidation, emotional memory processing, and the integration of new information with existing knowledge. The male REM advantage during early adulthood may support generally stronger procedural and implicit memory consolidation. Women and men run on slightly different internal clocks. The intrinsic circadian period, the endogenous length of the day-night cycle measured in controlled conditions, is approximately 6 minutes shorter in women than in men. Women's internal clocks tick slightly faster. 6 minutes seems trivial. It's not. It means women are more sharply out of alignment with 24-hour social schedules. The resulting circadian misalignment is estimated to be approximately 5 times larger in women than in men under standard schedules. And circadian misalignment impairs memory consolidation by disrupting the timing of sleep stages relative to circadian phase. Women secrete melatonin earlier in the evening than men, consistent with their earlier preferred sleep timing, and the amplitude of the melatonin secretion peak is larger in women. Higher melatonin amplitude is associated with sharper circadian signal strength. The earlier melatonin onset in women means they often go to bed closer to their melatonin peak under standard work schedules, supporting more efficient sleep initiation and deeper early night slow-wave sleep, which is the phase most critical for declarative memory consolidation. The well-known age-related decline in slow-wave sleep does not follow the same timeline in both sexes. A study found that in men, slow-wave sleep declines substantially between ages 20 and 40. In women over the same period, this decline is largely absent. The sex difference in slow-wave sleep may become largest between ages 30 and 40. Men in their 30s have measurably less deep sleep than they had at 20. Women in their 30s have maintained their slow-wave sleep levels. Given what slow-wave sleep does for memory consolidation, this male decline represents a biologically scripted reduction in consolidation capacity during peak career and family formation years. The female advantage in slow-wave sleep extends beyond time spent in that stage to the amplitude of the slow waves themselves. A systematic review found that approximately 75% of studies comparing slow-wave amplitude between sexes report higher values in females. Higher amplitude reflects larger, more synchronous neuronal oscillations across cortical regions, a signature of deeper neural quiet and more coordinated memory transfer between the hippocampus and the cortex during the night. The distribution of slow-wave activity across the scalp also differs. Women show more slow-wave activity over posterior and central cortical regions at bedtime, regions responsible for perceptual and motor integration. The female slow-wave advantage isn't diffusely distributed across the whole brain. It's concentrated in regions with strong inputs from daytime experience, potentially facilitating more efficient consolidation of the sensory content from the preceding day. A study of 40 participants, 20 men and 20 women, tested what happens to memory after a 1-hour daytime nap. All participants studied pictures with colored backgrounds, allowing the researchers to test not just whether they remembered the pictures, but whether they remembered the source context, the color of the background. For women, the nap improved source memory retention compared to staying awake. The effect was statistically strong. For men, napping had no benefit on source memory and actually resulted in worse performance. Item memory, just recognizing the pictures, was unaffected in both sexes. The nap specifically helped women bind items to their contexts, and it specifically failed to help men do the same. Within the nap group, a sex-specific correlation appeared. For women, improvement in recollection, the rich, detail-laden form of memory, correlated with the duration of stage 2 sleep. In men, no sleep stage correlated with any memory measure at all. Stage 2 sleep is characterized by sleep spindles, brief bursts of rhythmic neural activity linked to hippocampal neocortical memory replay. Female memory consolidation during naps appears specifically tied to this spindle-rich activity. Not all sleep memory effects differ between the sexes. Sleep following mental rotation training improved performance regardless of sex. Both men and women benefited equally from overnight consolidation of spatial skills. Procedural skill consolidation appears to be a more sex-neutral process. It's episodic memory binding, the linking of items to contexts, details to emotions, facts to sources, where the divergence is most pronounced. For declarative memory tasks, some research found that men with naps showed improved performance, while women showed nap-based improvements only during the mid-luteal phase of their menstrual cycle. The mid-luteal phase is characterized by elevated progesterone and a secondary estrogen peak, hormonal conditions that promote slow-wave sleep and enhance hippocampal encoding. So the popular advice to nap for better memory may be sex-dependent and cycle-dependent, most reliably true for men regardless of hormonal state, and for women, specifically during the hormonally receptive luteal window. While men experience a gradual testosterone decline over decades, women undergo a more abrupt hormonal transition at menopause. Hot flashes, night sweats, altered progesterone levels. These substantially disrupt sleep continuity, reduce slow-wave sleep, increase nighttime awakenings, and shift circadian timing. Because slow-wave sleep is the primary stage for declarative memory consolidation, menopause-related sleep disruption represents a direct and measurable threat to memory health, one that compounds the hippocampal connectivity changes and estrogen depletion effects we discussed earlier. Now let's look at what happens to memory across the longer arc of a lifetime, and at the diseases that threaten it. Alzheimer's disease is disproportionately a women's disease. Almost 2/3 of Americans living with Alzheimer's are women, an estimated 4.2 million women compared to 2.7 million men. The lifetime risk starting at age 45 is 1 in 5 for women, 1 in 10 for men, a twofold difference that persists even after accounting for women's greater longevity. A meta-analysis calculated women's odds ratio for Alzheimer's at 1.56 relative to men. This disparity reflects biological vulnerability, the loss of estrogen's neuroprotective effects at menopause, combined with the fact that the APOE4 genetic risk allele confers greater risk in women than in men who carry the same variant. Long-term prospective studies have examined whether hormone replacement therapy can reduce Alzheimer's risk. One 16-year longitudinal study, led by Kawas and colleagues, found that hormone therapy was associated with a 54% reduction in Alzheimer's disease risk. Over half the risk gone. That finding reshaped how scientists thought about estrogen. It was no longer just a reproductive hormone. It was a neuroprotective agent. But timing matters profoundly. Researchers have established what they call the critical window. Women who begin hormone therapy around the time of menopause, within approximately 10 years of their last menstrual period, show reduced dementia risk. Women who start much later, when Alzheimer's pathology may have already begun, don't see the same benefit and may actually face increased risk. This explains a confusing finding from the Women's Health Initiative memory study, which enrolled women averaging 65 years old, many years past menopause. That study found increased rather than decreased dementia risk with hormone therapy. The participants had missed the window. The biological mechanism behind the critical window is the gradual disappearance of estrogen receptors from the aging brain. After permanent estrogen loss, estrogen receptors in the hippocampus and prefrontal cortex gradually vanish. Once they're gone, administering estrogen can't restore the synapse-building, neuroprotective, and BDNF-stimulating effects that depend on those receptors. The cellular machinery is simply no longer present to respond. A clear clinical imperative. To preserve the estrogen-responsive memory system, hormonal support must begin before receptor expression has declined. A 2024 study in cerebral cortex revealed a specific functional deficit in postmenopausal women. They lose the normal ability to suppress default mode network hippocampal connectivity during active memory encoding. In younger women with intact estrogen, the default mode network appropriately quiets down as the brain shifts into encoding mode, freeing hippocampal resources for new information. Postmenopausal women fail to make this shift. Their default mode network stays active during encoding, creating interference. Not structural damage, a dysregulation of the attentional gating that normally protects new memory formation from noise. One of the most clinically important findings about female memory concerns what happens when cognitive decline begins. The well-documented female advantage in verbal episodic memory, present in cognitively healthy adults, is entirely absent in patients with mild cognitive impairment. In healthy adults, women outperform men on verbal memory tasks. In mild cognitive impairment patients, this advantage vanishes completely. The implication is troubling. Because women score higher at baseline, they can sustain more neural damage before falling below diagnostic thresholds. They appear normal for longer. By the time they're finally diagnosed, the disease is more advanced. Standard diagnostic tools, calibrated on sex-neutral norms, may systematically miss cognitive decline in women. While women bear the greater Alzheimer's burden, men show a distinct pattern of age-related cortical thinning. Greater decline in the cuneus, lingual gyrus, parahippocampal cortex, and paracalcarine cortex, all regions critically involved in visuospatial memory. Two sexes, two aging trajectories, men with more prominent visuospatial circuit atrophy, women with more prominent hippocampal vulnerability to disease. A cross-sectional analysis of episodic memory across the lifespan found that women show a more pronounced decline in their 50s compared to men of the same age. This decade corresponds precisely to perimenopause and early postmenopause. The timing matches the precipitous drop in estrogen. It matches the imaging data showing hippocampal functional disruption beginning in the perimenopausal period. Many women first notice memory difficulties during perimenopause, not as a subjective illusion, as a real, measurable, hormonally driven decline. Sex differences in neurodegeneration extend beyond Alzheimer's to Parkinson's disease, where the prevalence pattern reverses. Men face approximately twice the risk of being diagnosed with Parkinson's compared to women. But women who do develop Parkinson's show faster disease progression, greater motor decline, and worse quality of life outcomes after diagnosis. Greater male prevalence, greater female severity, opposing patterns suggesting that sex hormones influence both the probability of disease initiation and the speed of decline through different mechanisms. The biological machinery of Alzheimer's pathology, tau tangles, amyloid burden, the APOE epsilon 4 allele, appears to affect women's brains more severely than men's with equivalent pathological load. Total tau and amyloid beta levels in women are associated with steeper cognitive decline and greater hippocampal volume reduction. Women in the prodromal stage of mild cognitive impairment show faster deterioration and more severe biomarker trajectories than men carrying the same genetic risk. The CA1 region of the hippocampus, the very region where estrogen drives the most dramatic synaptic growth, is also the first region severely affected by neurofibrillary tangles. The site of greatest plasticity becomes the site of greatest vulnerability. Let's shift now to a different kind of brain change, not one caused by disease, one caused by new life. Pregnancy produces documented, lasting structural changes in the maternal brain. First-time mothers undergo extensive gray matter volume reductions in the anterior and posterior cortical midline, including the medial prefrontal cortex, posterior cingulate cortex, and precuneus. These reductions are still present more than 2 years after giving birth. These structures form the core of the default mode network, and the regions of gray matter reduction overlap with the theory of mind network, the brain system responsible for attributing mental states to others. This is not cognitive deterioration. This is neural specialization. The brain is pruning itself for a specific purpose. Efficiently reading the mind of a pre-verbal infant who can't describe hunger, pain, or fear in words requires exactly the kind of rapid social inference supported by the theory of mind network. The gray matter changes may represent experience-dependent synaptic pruning, a refinement of circuits toward faster and more efficient infant-attuned social cognition, not a loss of capacity, a sharpening of function. And the degree of brain change correlates with the strength of maternal bonding. More change, stronger bond. The structural changes of new motherhood are not all reductive. A study tracking gray matter from 2 to 4 weeks postpartum through 3 to 4 months postpartum found that mothers show gray matter increases in the prefrontal cortex, parietal lobes, and midbrain. These regions support planning, attention, sensory integration, and reward processing, precisely the capacities needed for newborn care. And mothers who perceive their baby more positively, rating the infant as special, showed greater gray matter augmentation in the hypothalamus, substantia nigra, and amygdala, regions central to bonding, reward, and protective vigilance. How you feel about your baby physically changes the structure of your brain. Fatherhood also restructures the brain. New fathers show gray matter increases in the hypothalamus, amygdala, striatum, and lateral prefrontal cortex, while showing decreases in the orbitofrontal cortex, posterior cingulate, and insula. The magnitude of brain change in fathers is approximately half that of mothers, but the convergence on overlapping reward and bonding circuits in both parents suggests the neural architecture of parenting is at least partially shared between the sexes, even if the hormonal drivers differ. A 2026 study tracking women across first and second pregnancies found that the second pregnancy produces less pronounced changes in the default mode network compared to the first, but it shows stronger alterations in the dorsal attention network and somatomotor networks. Some first pregnancy specialization is retained, not rebuilt from scratch. New adaptations in attention and motor control emerge for the second child, perhaps reflecting the demands of managing a mobile toddler while caring for a newborn. The largest study of pregnancy brain changes to date, involving 127 women, confirmed that more pronounced brain changes correlate with stronger infant bonding. Now let us move towards something that may sound like it belongs in a different kind of story. One of the most dramatic demonstrations that memory decline with aging is at least partially reversible came from experiments in which old mice and young mice were surgically joined, so they shared a circulatory system, a technique called heterochronic parabiosis. In a 2014 study published in Nature Medicine, led by Villeda and colleagues, aged animals exposed to young blood showed reversal of pre-existing brain aging at every measurable level. At the molecular level, changes in gene expression related to synaptic plasticity. At the structural level, increases in dendritic spine density in hippocampal neurons. At the functional level, improved long-term potentiation. At the cognitive level, better performance on learning and memory tasks. The reversal applied to damage that had already occurred, not prevention of future decline, but actual reversal. Some components of brain aging are not permanent. They are actively maintained by what's flowing through the blood. The molecular mechanism behind this reversal was partially identified. In the hippocampi of old mice exposed to young blood, a transcription factor called CREB was reactivated. CREB is central to long-term memory formation. It's required for the late phase of long-term potentiation, for the consolidation of lasting memories, for the expression of BDNF and other plasticity genes. Young blood doesn't repair the aged brain through some mysterious vitality. It reactivates a specific molecular switch, CREB, that the aged brain still possesses but has stopped using. The experiment has a mirror image. When young mice were exposed to old blood plasma, the effects were harmful. Hippocampal neurogenesis declined. Synaptic plasticity was impaired. Learning and memory performance worsened compared to young mice receiving young plasma. Old blood doesn't merely lack rejuvenating factors. It contains active inhibitory factors that directly damage the young brain. A bidirectional model. Aging produces both a decline in helpful molecules and a rise in harmful ones. The search for the harmful factor in old blood led to a molecule called CCL11, also known as eotaxin, a chemokine primarily known for its role in allergic inflammation. CCL11 plasma levels increase with normal aging in both mice and humans. Levels in cerebrospinal fluid also rise with age. Direct injection of CCL11 into young mice reproduced the neurogenic suppression seen with old plasma, reducing newborn neurons in the dentate gyrus. And a neutralizing antibody against CCL11 rescued the effect. A single molecule from aged blood, sufficient to impair both neurogenesis and cognition in a young brain. Looking beyond blood to the brain's own internal fluid, young cerebrospinal fluid, when infused directly into aged mouse brains, improved memory function and boosted the proliferation of oligodendrocyte progenitor cells in the aged hippocampus. The active ingredient was identified as fibroblast growth factor 17. Direct infusion of this single factor was sufficient to induce cell proliferation and long-term memory consolidation in aged mice. Blocking it impaired cognition in young mice. The fluid that bathes your brain carries its own set of age-dependent signals, and those signals can be manipulated. Research from Tony Wiscore's laboratory at Stanford showed that the young blood effect crosses species boundaries. Plasma collected from young humans improves neural stem cell activity and memory performance in aged mice. Plasma from elderly humans makes young mouse brains more inflamed. The underlying molecules and pathways appear to be evolutionarily conserved. What works in rodents may have relevance in human brains. 3 independent research groups, working on different questions through different experimental approaches, converged on the same molecule, platelet factor 4, PF4. One group from Saul Villada's lab came at it from the young plasma direction. Another from Dina Dubel's lab came from the clotho longevity hormone direction. A third from Tara Walker's lab at the University of Queensland came from exercise physiology. All 3 found that PF4 was the common factor transferring rejuvenating effects from blood to brain. When 3 independent paths lead to the same destination, the destination is probably real. PF4 was injected into 20-month-old mice, roughly equivalent to elderly humans, every other day for about 24 days. The aged mice showed measurable improvements across multiple independent memory tests: novel object location, contextual fear conditioning, active place avoidance. PF4 treated aged mice performed substantially closer to young mice than to untreated aged controls across all measures. And when the researchers eliminated newborn neurons in the hippocampus using diphtheria toxin, the memory benefits of PF4 disappeared completely. PF4 works through promoting the birth and maturation of new hippocampal neurons. Remove those neurons, and the benefit vanishes. At the cellular level, PF4 increases the number of immature neurons in the dentate gyrus of aged mice. It promotes neuronal survival and differentiation, guiding cells out of the proliferative cycle and into mature neuronal identity. In mice genetically engineered to lack PF4, baseline hippocampal neurogenesis dropped well below normal levels. And exercise, which normally boosts neurogenesis, no longer worked. PF4 isn't just sufficient for the neurogenic response to exercise. It appears necessary. Platelets, the tiny cell fragments best known for blood clotting, turn out to be key players in keeping the brain young. An upstream activator of PF4 has been identified: clotho, the longevity hormone produced in the kidney. Administering clotho to mice elevated plasma levels of PF4. Because clotho itself is too large to cross the blood-brain barrier, it acts through platelets as intermediaries. Clotho activates platelets. Platelets release PF4. PF4 enters the brain and stimulates hippocampal neurogenesis, a chain of signaling from kidney to platelet to brain, connecting longevity to memory through the blood. Exercise is the most powerful known way to generate youthful blood factors that improve brain function. Research from Wiscore's group showed that exercise triggers the liver to produce an enzyme called GPLD1, which amplifies the production of neuroprotective factors in the blood. Blood from exercising aged mice, when transfused into sedentary aged mice, improved hippocampal function. And GPLD1 was identified as a key factor mediating this benefit. Exercise functions as a natural young blood generator, producing endogenous rejuvenating molecules that mimic some of the effects of sharing a young circulatory system. A conceptual shift has emerged from this body of research. The age-related decline in new neuron production isn't primarily caused by the disappearance of neural stem cells. The cells are still there, in substantial numbers. They've been silenced by inhibitory blood signals. When old blood is replaced or diluted with young blood, the same dormant cells resume their work. The brain's capacity to generate new neurons in old age hasn't been destroyed. It's been suppressed. And suppression, unlike destruction, can potentially be reversed. The scope of the young blood effect reaches far beyond neurons. Single-cell RNA sequencing across 20 organs showed that young blood reversed aging signatures in nearly all cell types across all 20 organs examined. Mitochondrial genes, the energy-producing machinery inside cells, were among the most consistently responsive, showing rejuvenation across cell types after young blood exposure. And interestingly, young blood and exercise target overlapping but distinct cellular populations. Young blood primarily rejuvenates neural stem cells. Exercise primarily rejuvenates oligodendrocytes, the cells that insulate neural wiring. Growth differentiation factor 11, GDF11, is another molecule found in young blood with neurogenic properties. Administration of GDF11 to aged mice increases neural stem cells in the dentate gyrus and improves memory task performance. Examination of neuronal activity markers confirmed that GDF11-treated neurons showed enhanced firing. GDF11 levels decline with aging. Restoring them to youthful levels produces structural and functional brain rejuvenation. And PF4 does more than promote new neurons. It also reduces neuroinflammation in the aged brain. PF4 decreases the expression of pro-inflammatory genes in the hippocampus and reduces the activation of microglia, the brain's resident immune cells, which in their reactive state prune synapses and drive cognitive decline. PF4 protects memory through two simultaneous mechanisms: building new neurons and reducing the inflammation that destroys existing ones. These rejuvenation findings work in both male and female subjects. The blood composition axis of cognitive aging appears to be, for once, sex-neutral. a rare convergence in a field otherwise defined by persistent differences between the sexes. Now, as the narration approaches its final stretch, let us hold still for a moment. The discovery that circulating blood factors can reactivate memory function in aging brains is among the most quietly important findings of recent neuroscience. Stem cells reigniting, inflammation receding, neurons firing with renewed activity, not through transplanting new cells, but through restoring the chemical environment that allows existing cells to do what they were always capable of doing. Consider this. Every memory you hold has been shaped not just by what happened to you, but by the particular chemistry of your own brain. A chemistry influenced by your sex, by your hormones, by the quality of your sleep, by your age, by what's flowing through your bloodstream right now as you listen to these words. And right now, your brain is holding this very episode in short-term memory, preparing to decide what to keep and what to let dissolve by morning. The male and female brain are far more alike than they are different. The vast majority of neural architecture is shared. The same regions, the same neurotransmitters, the same fundamental processes. But the subtle variations in how those shared structures encode and retrieve memories reveal a quiet complexity. Women bring a more emotionally integrated system that excels at episodic autobiographical memory, object location associations, and emotional face recognition. Men bring a more spatially organized system that excels at three-dimensional rotation, large-scale navigation, route-based memory, and abstract visual pattern recall. Neither system is superior. Each has strengths the other lacks, and both remain plastic, trainable, changeable throughout the whole of a life. The brain doesn't remember perfectly. It remembers usefully, shaped by biology, by emotion, and by the slow rhythms of each night. The narration grows quieter now. Somewhere in the darkness tonight, your neurons are preparing for their work. The hippocampus will replay fragments of the day. The slow waves of deep sleep will wash across the cortex, carrying information from temporary storage into longer-term networks. Some of what you heard tonight will be kept. Some will be gently released. The brain will make its own choices, silently, while you rest. Memories form, memories change, memories soften over time. And in every resting brain, male or female, young or old, the same ancient process unfolds, the same slow oscillations, the same quiet transfer of experience into lasting knowledge. Stars drift overhead, the planet turns. Somewhere a neuron fires, and somewhere inside you a memory is being made. Sweet dreams, and we will see you next time on Bedtime Science.